neurodegenerative disease. And I mentioned that particularly we are focusing on one disease. Did I talk about Huntington's disease before
Yes, ma'am.
in my last
Yes,
class?
ma'am.
Okay.
Yes, ma'am.
Know that this polyglutamine disease, the name poly comes from expansion of glutamine, which is a triplet repeat CAG. Okay, so in normal human being, the glutamine number is below 35, but the moment there is onset of the disease, this number is going to increase beyond 35. Okay, and this is one of the thing which I showed you, I think, in my previous class, that mainly neurons are getting degenerated, and therefore it is called neurodegenerative disease. Okay. On the right hand side, this is the brain from Huntington patient, postmortem brain, and left hand side is the normal brain. And at bottom, you can see a patient who was suffering from HD, and this is almost terminal stage for the person. It's very, very interesting that some of the people, they survive quite long despite of the devastating symptoms of this disease. You must be aware that even Stephen Hawking, he survived for a very long time. But as most of the patients, they die quite early because they are not able to perform their day-to-day activity, and along with day-to-day activity, they are not able to even eat properly, and they become like cachexia. आपको पता है जब पूरा muscles are getting lost, that is the cachexia stage. Okay, so they become really lean and thin. वो आपको यहाँ पे भी नजर आ रहा है patient में. So I think I told you about this part as well, and I told you that finally we concluded that indeed these flies can be used for uh, understanding the various mechanisms involved in Huntington's disease, and in addition, they can be very useful for screening drugs. Ultimately, this is the aim, that flies should, the patients should get the drug which has least side effect, okay, and they are not suffering from the symptoms which I mentioned before. So, just to, because I want to finish everything, there was a significant concordance of drug response in flies and mice. Okay. So, whenever we try to narrow down the drugs using fly as a model organism, I might have told you that pharmaceutical companies, they were sending us drugs so that it can be tested in the fly model, and it was all blind testing. So we did not know what type of drug it is. So we were testing drugs, and the moment the drugs are narrowed down, क्योंकि एक कंपनी 500,000 drugs बनाती है, लेकिन they need a system by which it can be tested quickly, and Drosophila served really, really well that way. So out of 500, the number would be narrowed down to 5 or 6, depending upon its effect in HD fly model. And then it was validated in the mammalian system using mouse as a model system. Once it is, it works very well in the mouse, then it goes for the clinical trial. And all of you must be knowing that there are clinical trials, so it is going to pass one clinical trial number 1, then only it will enter into the second one. So what we observed, that whatever drugs we tested in mouse, and it turned out to be positive in terms of HD treatment, they also worked very well in the mouse model. Okay, we could also identify the targets of these drugs, because we were involved in understanding about the mechanism, molecular mechanism of these drugs. And as you can see in the fourth panel, that whatever could rescue in fly, that also rescued in mouse and in human being as well. As far as Saha is concerned, as far as system is concerned, they also passed all the clinical trials and they are under the treatment regime. So, but before I stop talking about the pharmaceutical drug, another interesting part which we, performed in our lab at Delhi University. So maybe I told you before that I wanted to test many phytochemicals because our country is full of herbs and really miraculous herbs. So we tested multiple herbs, and one of the most promising one is curcumin. जो हल्दी आप लोग खाते हो, है ना, घर में हल्दी डालते हैं ना खाने में, हर कुछ खाने में, या फिर कई बार होता है अगर you are hurting yourself, then your parents will tell put haldi there, okay, because of multiple properties this curcumin has. So it turned out that curcumin is the most promising treatment regime for Huntington's disease. As mentioned, we tried so many drugs. We tested more than 5,000 drugs, but curcumin is like a charm and it doesn't have side effect as well. So with least side effect and because of multiple targets, it really works well. Okay. So if I'm changing the slide, can you see? I'm on the next one.
No,
Because again,
ma'am.
I
No, ma'am.
No, ma'am, it's still the
so I
table.
don't have a
Yes,
presentation.
ma'am.
Right now.
So this was all about the basic thing. We are working towards the molecular mechanisms. We are working towards the, trying to understand if post-translational modifications are involved in this disease. And there are many things which are going on. But anyway, the point is you can think about the most complicated uh, technique, okay, any technique. कई स्टूडेंट्स कहते हैं हम लोग अभी वी वर कंडक्टिंग पीएचडी वाइवा फॉर लास्ट 2 days, आप लोगों की क्लासेस इसीलिए नहीं हो रही थी, है ना, यू गॉट द मैसेज आल्सो, and the students will tell we will ask them which topic you are interested in. They would say molecular biology or molecular techniques. So, molecular biology and molecular techniques are not the questions, right? So, using fruit flies, multiple questions which are related to the molecular mechanism can be addressed. Okay, this is such a simple system. You can do whatever you want. Okay, so in addition to, we found very clearly that phosphorylation is one of the most possible post-translational modification which is leading towards uh, enhancement of disease. Okay, in addition to that, we also could clearly say that sumylation is one of the post-translation modification. So why I'm saying all these things? The simple reason is you have to understand that question should be proper and you can use the most simplest model organism like fruit fly to address your question. So as I said, I have been working on tumor suppressor genes during my PhD, then started working on neurodegenerative diseases. Um, but there was a student who was my MSc student as well, like all of you. She came to me, she said she wants to work on fruit flies, but she doesn't want to work on neurodegeneration. Okay, so I told her clearly that my lab is only interested in neurodegeneration, so you should better find a different place. But she was a very good student, and at the same time, she was quite adamant that she would like to work with fruit flies only. And there is no one in the department or even in Delhi University or other places who is considering fruit flies as a model organism. So she kept on telling me that I want to work on nanoparticles. And believe me, I was not at all interested because I had so many questions related to Huntington's disease. Um, but then, somehow it is, uh, there was situation when one of my childhood friends, she was working at University of Dallas. She was asking me all the time, can we test, and she's a physicist, so she was telling me we are synthesizing nanoparticles for targeted drug delivery and for medical applications, but before using them in the mainstream, we would like to test them in a model organism. So, will it be possible for you to collaborate with us? And I was always denying this. But when Akanksha, the student, came to me, and she was really hard on me, I would say. So then I spoke to Professor Prasanna Shah, who was there in Dallas, and then we started working on nanoparticles. So, why nanoparticles? Nanoparticles have multiple advantages. They are mysterious particles, but at the same time, it is very, very important for all of us to know that these nanoparticles are part of our day to day life. The plastic bottles we use, of course, the good ones, because nanoparticles are expensive. Then, the good plastic wares are going to have nanoparticles because nanoparticles give strength to plastic wares. They are also part of our, for several medical applications like imaging, MRI, whatever you name, it's there. It is also considered as one of the best way for targeted drug delivery. Okay, so but in addition to all these things, these nanoparticles, particularly silver and gold nanoparticles, they occupied huge space in cosmetic industries. Okay, so अपनी day to day life में तो है, even in toothbrush, you'll be surprised to know. So, cosmetic industry is full of nanoparticles, particularly silver and gold nanoparticles. But the limitation is right here. They are very useful, but at the same time, they are not evaluated properly for their dosage. And therefore, I am writing it right here that all the prescription drugs जो भी हम medicine वगैरह use करते हैं, they are being subjected to peer-reviewed laboratory trials. But drugs, cosmetics, and consumable with nanoparticles are not at all subjected to same degree of scrutiny. Not only that, the details of the nanoparticles are not even mentioned on the label. आपको पता है आजकल ये FDA का एक basic requirement है that ingredients has to be written, okay, and how much, what kind of compound is being added there should be there. So the name and how much, that is the requirement. Okay, so nanoparticles, even if they were incorporated, they were not mentioned on the label. FDA and doctors agree that they need to be tested. So now it is a mandate. So I was telling you about the cosmetic industry. This cosmetic industry is full of nanoparticles. So how they found their place in cosmetic industry? थोड़े समय पहले आप लोग सुनते होंगे कि most of the cosmetic products, they had vitamin C and vitamin E. Simple creams also. But the problem was that these cosmetics were not able to enter inside our skin. And if they cannot enter our skin, their effect is going to last not for very long time. Okay. So if you talk about the foundation, just an example, what happens? You put the foundation and you would like that you glow entire day. And this particular observation started when I visited US, and like malls here now in India also, there are small stalls everywhere for cosmetic products. So the girls over there, sales girls, they will call you. They will try to show you their product, and they will also like to apply the product on you. So one girl was saying, please come here, and I'm going to put a foundation. You are going to glow entire day. And by that time, we had started working on nanoparticle. So this is the story I am telling you about my own experience. And I went to that girl and I asked her, "What do you have in there? How can I glow entire day?" And she says that now we have started adding silver and gold nanoparticle. And the property of nanoparticle by name itself is very clear to all of you. These are very tiny in their size, less than 100 micrometer. Okay, and therefore anything which is less than 100 micrometer is going to be considered, or 100 micron is considered as nanomaterial. Okay, so because of their small size, they had the capability to get absorbed into the skin. And the moment gold and silver nanoparticle will be absorbed inside the skin, of course you are going to glow for whole day. So I asked her, can you please tell me how much is being added there? And she said, we don't know about that. I looked at the ingredient part, it's not there. And believe me, it was one of the best companies. You must have heard about Naturals. Okay, so I mean, even the good companies who use, all the ingredients after conducting research, they also did not mention how much is being added. L'Oreal is another example, which is right here. So you would not know how much nanoparticle is being added. So people initially were highly excited with incorporation of nanoparticle, but as the time passed by, the effect was very, very clear. So people were having problems on their skin. The nanoparticles which were incorporated into the lipsticks and other things, they were also having serious issues. And there was huge agitation in Western countries that this is the toxic beauty. Okay, so it should not be used without proper investigation. So there were guidelines from FDA and medical industry that there should be evaluation of nanoparticles before its incorporation into any of the product. Okay, so hopefully it is clear to you. And there comes our story now that this was our first objective, that we have to find out a proper dose of nanoparticle. Okay, because these are really very advantageous. Uh, just a minute, let me fix the charger here. Okay. So, yeah, so coming back now, these are miraculous. If you talk about the advantage, they are uncountable. And as far as medical industry is concerned, it's a boon for medical industry. ओके, जो भी कैंसर में इंटरेस्टेड हैं, जो भी न्यूरो डिजनरेशन में इंटरेस्टेड हैं, you can think, the one of the biggest property of the nanoparticle is they have they don't have blood brain barrier. They can cross even the blood brain barrier. Okay, so if you talk about encapsulating nanoparticles with certain drug and target it to a particular place, it's going to work like miracle, because rest of the normal cells will remain unaffected. Okay, so this was our first hypothesis, that look at we should try different dosage of nanoparticles using Drosophila as a model organism. And initially we started our work with silver nanoparticle, because silver nanoparticles have antimicrobial property, and they are most commonly used nanoparticles in consumer products. including cosmetics. And as I said, we looked into multiple, aspects in fruit flies. For example, we started looking into their reproductive capability, that if flies were fed with different concentrations, that means different dosage of nanoparticles, what happens to their egg laying capability. And as you can see very well here on the left hand side, these flies were fed for nanoparticles for 10 days. Okay. And I'm not going to go too much into the scientific details, but if you are interested, maybe we can talk about it later once we are done with our syllabus. So, the with the highest concentration, their egg-laying capability was significantly reduced. And then another question was whether the impact is going to get further pronounced if they are aged. So, what we found at day 10, uh, their egg laying capability was reduced, but when they were 30 days old, then also their egg laying capability was significantly reduced, which is also clear from this graph. आप अगर दोनों ग्राफ में जो भी बच्चे इंटरेस्टेड हैं वो ज़रूर नोटिस कर रहे होंगे कि फ्लाई की एग लेइंग कैपेबिलिटी एजिंग के साथ अपने आप कम होती है. If you compare this white graph with this one, this is 10-day old fly, this is 30-day old fly, their egg laying capability reduces almost to the 50%, right? But this egg laying capability, which is already reduced due to their aging, is further compromised by incorporation of silver nanoparticle in their food. Okay. However, our aim was not to figure out the toxicity or toxic behavior of nanoparticles. We were more interested to convey the message that if a proper dose is being used, then they are going to be harmless. And you can see from this study itself that there was a dose which was at lower concentration, that is 0.05%, I think. Yeah. Is that right? No, it is 0.01. Then it was quite comparable to the control wild type flies. Okay. Now, when these eggs developed into the larvae, it was really shocking for us to see that the larvae at higher concentration were not able to grow at all. They were so small in size, it was a mixed population. So some of the larvae were smaller in size as compared to the control, but some of them were really really weak. Because अब population में भी क्या देखते हो, आपने ये सुना है कि सबका every individual has its own tolerance level. Okay, same is true for the flies. So some of the larvae could develop up to this, they could grow up to this level, but some of them could not grow at all. They were like first instar larvae. Okay, so at the same time, at lower concentration, we could see that they were quite comparable to the control. So it clearly suggested that silver nanoparticle has impact on larval growth and development as well. We also looked into the survival, and I would ask you to pay your attention to the these graphs, which are right here. Don't look at the white ones. White ones are the control, and these are the experimental ones. Kind of apricot color, I would say. So we could clearly see that they had problem in their survival. And when we talk about the survival, we looked at different developmental stages. This is the stage which is pre-pupal stage. जब मैंने आपको explain किया था life cycle, आपको बताया था कि क्या pre-pupal stage होती है. So larvae से next step. Okay, so we could see that the larvae could not form a prepupal stage. Later on, when they became pupa, then also the pupal rate was significantly reduced as compared to the control. And if you talk about the eclosion level, please focus on my cursor at 0.5%. They very few flies could hatch out, and the moment they hatched out, they were dead. Okay, so this was the most toxic dose, I would say, but the safe dose was always there. Look at this apricot colored bars, okay, and compare it with the control, which is in white color. Now, from some of the concentrations, which were intermediate concentrations, flies also hatched out, but I would like to draw your attention to the upper panel where you can see these are the pupae or pupa, where metamorphosis has already taken. And once the metamorphosis happens, you can see the color of the pupa becomes blackish, right? And um, as the dose of silver nanoparticle was increasing, this pigmentation was getting compromised. Can you differentiate between all these concentrations, 0.01 to 0.1 and then 0.5? Yeah. So as the concentration was increasing, their pigmentation was getting reduced. And the flies which came out or hatched out from these pupa, they were also having significant problem in their pigmentation. Compare these two larvae with these two, left hand side versus right hand side. So the control is totally normal, which was not supplemented with silver nanoparticle, and you can see very nice pigmentation. You can also see abdominal segments, but when higher dose of silver nanoparticle was administered, they had problem in melanization, and therefore they could not form the pigment on their body. You cannot see these abdominal segments either. However, the lower concentration, which is 0.01%, flies were quite comparable to the control. Okay, so in addition to their survival, their phenotype, we also looked into their behavior. So we don't care that much about our appearance, but at I mean, we do, but not up to the level. But we do care about our performance, whether we would be able to live our day-to-day life normally or not. And for flies, it is the climbing behavior because they are flies and they are negatively geotaxic. They love to climb, and what we found that these flies had significant, which were fed with higher concentration of silver nanoparticles, they were not able to climb at all. Okay, and in addition to their climbing, we also looked into the crawling behavior of the larvae. They had significant trouble in their crawling and climbing behavior. And this is about the survival. Yes, someone was saying something. Please.
Yes, ma'am. Ma'am, actually, I wanted to ask that can we standardize the same result for human beings as well? Because the duration of lifespan for Drosophila is quite less as compared to
Yes.
human beings.
Right.
So, how can we standardize these results? Like you said, it's a model organism. So,
Right.
how can
Yes. So the message what we are conveying using this model organism, that the dose has to be evaluated. Okay, as you said, that is very rightly said. बहुत छोटा सा, they live for 3 months, right? These fruit flies live for maximum for 3 months, but we live for 60 years. So whatever observations in terms of phenotype or behavior is going to happen in human being is will be taking much longer as compared to flies.
yes, ma'am.
So, yeah, so this model organism is clearly suggesting that higher concentration of silver nanoparticles, or in other words, just to generalize, that silver nanoparticle, if not evaluated the dose beforehand, can have toxic, severe toxic effects. And that can be, this message can be clearly used in human beings. क्योंकि जो भी लोग मैमेलियन सिस्टम में काम करते हैं, फॉर एग्जाम्पल वी, there was no study. So what we have to do, in vitro studies are there. From in vitro study, we know how much concentration was used. And all of you know, for in vivo study, you need higher concentration. So we had to standardize concentrations, before showing this data or publishing this data. So do you think we tried only 5 concentrations? Answer is no. We started with a really high concentration, and we found that none of the larvae were surviving, and then we tried to dilute the concentration. So same can be done for the mammalian system, but the message is very, very strong, that look at the flies. These are the model organisms, and they are used with nanoparticles without evaluating the dosage. Then it's going to have serious issue in human beings. और आपको मैंने दिखाया भी, आपको मैंने वो there were agitations all around the world, और वो मैंने आपको दिखाया. The impact took a very, very long time, but it is very serious. Okay, and flies are clearly telling कि अगर आपने सिल्वर नैनो पार्टिकल या गोल्ड नैनो पार्टिकल बहुत ज्यादा कॉन्सेंट्रेशन में यूज किया, तो पिगमेंटेशन पे अफेक्ट होगा, ग्रोथ पे अफेक्ट होगा. एंड यू मस्ट हैव सीन देयर bellies also. I'm not going to tell you each and everything what we did related to nanoparticles. Time भी नहीं है ना? That is the limitation. But we published a paper very recently that देखो एकदम shrink की हुई belly है. So the lipid, protein, glycogen, whatever you name, the sugar, everything is getting compromised if these nanoparticles are in abundance. So अब हम लोगों ने क्या किया ह्यूमन बीइंग ने, using the brain, that yes, there are so many proper goods. These consumer goods, for example, plastic wares, they are going to last for very long. But what we are doing, अगर हमने पानी की भी बॉटल आप यूज करो, तो क्या होता है? धीरे 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 धीरे, very slowly, this poison is going to enter inside us. I'll not go into that detail, but अभी आप लोग ये सुनते होंगे कि कैंसर हैज इन, दूर की फैमिली हो, पास की फैमिली हो, people are suffering from cancer. Why? Very simple. We have seen nanoparticles. Nanoparticles are one of the culprit. But at the same time क्या होता है, हम लोग के घरों में हर घर के ऊपर प्लास्टिक टैंक्स हैं, सिंटेक्स या जो भी कंपनी हो. ओके, and with heat, all of you know what is happening there. We are drinking that water, and ultimately the consequence is right there. पहले आपके ग्रैंड पेरेंट्स से अगर आप पूछोगे, जिनका भी घर होता था या जिस भी घर में रहते थे, वहां पे कंक्रीट के टैंक्स होते थे. यूवी का इफेक्ट ऑन प्लास्टिक, मन हो तो जरूर पढ़ना. नैनो पार्टिकल का इफेक्ट जरूर पढ़ना. बट आई एम सिंपली ट्राइंग टू टेल यू, दे आर रियली रियली टॉक्सिक. ठीक है? So coming to the next thing, we looked into the reproductive ability. I told you about the egg-laying behavior, but look at this particular picture. What is happening here? This is the ovary of fruit fly. Extreme left-hand side is the control. Okay. Next to it is a safe concentration, and then these are the higher concentrations जिसका effect आपने पहले भी देखा. Now the question was whether the flies which are having food with nanoparticle, they are having problem in releasing their eggs, or they have problem in storing their egg, or third question is what is happening to the development of the ovary, and answer is right here. These are beautiful dissections done by, Akanksha again. And Akanksha finished her PhD long back. She's in US doing her postdoc right now. But look at the dissection, the kind of development in the ovary. Your left hand side versus right hand side. Dekho. Ovary is not getting okay. In addition to that, number of eggs are very less. यहाँ पे देखो, इसको देखो, इसको देखो. But we were even surprised to see that there are lesser number of eggs, but their morphology is completely distorted. Look at the shape of the eggs. ये जो दोनों तरफ से nicely pointed eggs होते हैं, they became like a tiny square or small balloon kind of eggs. और ये जो होगा तो all of you know very well they are not going to be fertilized eggs at all. Okay, so out of 100 eggs, very few of them are going to give rise to larvae. Okay, adult occlusion, indeed, it was highly compromised at higher concentrations. Look at these two. Okay, again, apricot colored bars, and significance is right here. And this gets further pronounced with, uh, aging. So, on the caption, you can see that I'm writing F2 progeny. What should be the message here? Using fruit fly as a model organism, you can also observe transgenerational effect. So what is transgenerational effect here? F1 progeny was supplemented with silver nanoparticle. Listen to me very carefully. Okay. F1 progeny or parental progeny was supplemented with silver nanoparticle, but F2 progeny was not at all supplemented with silver nanoparticle. ठीक है, और जो भी abnormalities मैंने अभी तक आपको बताई हैं, वो सब abnormalities, even F2 progeny not seeing silver nanoparticle had it. Okay. So repeating again, silver nanoparticle was supplemented to parents. Their progeny came out. Okay. They were not exposed to nanoparticle at all. But same impact like their parents in terms of their behavior, in terms of their egg laying ability, or ovary formation, is going to be maintained in generations to come. Is it clear? So for example, if I am exposing myself to very high concentration of nanoparticle, my kids will have to face the consequence, right? Because there is transgenerational, it has been observed in human beings as well. और क्या सिल्वर नैनोपार्टिकल और गोल्ड नैनोपार्टिकल ही सब कुछ करते हैं? आंसर is no. One is which are being synthesized. We are talking about the synthesized ones. And the synthesis is going to depend upon the interest of the scientist. Interest means the question the scientist wants to raise. but there are natural sources as well. दिल्ली में अभी सुना था ना कि pollution has crossed the limit. So these pollutants also carry nanoparticles. Anything which is less than 100 nanometer, and they can enter inside us or we can ingest them, they are nanoparticles. Okay, and you can very well imagine that if too much exposure is being caused to nanoparticles, then it will have consequence in future generations to come. Okay, so let us, yes.
I have a question. If certain low dosage of nanoparticle is given to one progeny, then and this is constant, that in every progeny we are giving low concentration dosages. Can we see a kind of slow poisoning in that case?
No, 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 no. That we did not observe, not at all. It at least in a fly model, I can say this lower concentration, which I consider as safe concentration, it did not have any transgenerational effect. It did not have any effect on the flies who were exposed to it. So lower concentration is safe.
Okay, ma'am.
Yeah. Another thing which I am not showing you, but you must be seeing P plus, L plus, this kind of abbreviations there. So we were also interested if these nanoparticles have impact on a particular developmental stage, or all developmental stages are prone towards the effects. Okay. And P stands for adults, that means parents, and L stands for larvae. Okay. So we wanted to see कि अगर जैसे अगर human being की बात करो तो throughout their life, if a child is getting exposed to nanoparticle versus parents, when they grew up, they are getting exposed to nanoparticle, and we clearly found that if exposure is done from larval stages onwards, then the effect is much pronounced. It's not only time, because that question also we tried to address whether it is the longer time. Answer is no, because if you are very young, your tolerance level is low, your immunity is low, no, and you are getting exposed to nanoparticles, then it will have significant effect on your health as compared to the adults. Okay, but it's okay, you cannot understand each and everything in one class. So as I said, these flies were very meek, and all these questions what I am telling you, they are coming from minute observation only. So one day Akanksha came to me, she said, ma'am, these flies do not fly, they do not even climb, and they were very tiny also. So when body weight is one of the important measure for the metabolic dysregulation. Okay. In addition, they were also deficient of energy, and that's why they were not able to climb properly. So we started looking into the body weight of the flies which were exposed to a higher concentration. And as you can see again with 3 stars, there was significant reduction in their fresh weight. If we quantitated, we evaluated total lipid level in these flies, and of course, the lipid level was significantly low in the flies which were exposed to higher concentration of nanoparticle. In addition to that, we also tried to monitor whether lipid is formed or not, and therefore we monitored subcellular localization of the lipid in the fat body of the flies. Long back when I started my class, when you joined, I told you Drosophila fat body is homologous to human liver. Okay, So these are the pictures of Drosophila fat body, and in the fat body there are lipid droplets. Lipid droplets are the places where lipid gets accumulated, and you can see them in red. Okay, so this is the control fly, which was not exposed to silver nanoparticle. You can see very nice lipid droplets which are shown in red. But the moment flies were exposed to higher concentration of nanoparticle, that is 0.05%, the fat body cells were getting affected, lipid droplets were getting affected, and the lipid in them. But look at their morphology. And there is a entire molecular process behind formation of lipid droplets. They were appearing as if two lipid droplets are fusing with each other. Okay, so yeah, and as I was telling you, this is a wonderful model organism which clearly suggests that you need to have a proper question is there to answer your most complicated question without taking too much time. Okay, so if you have any question, you can ask me.